I begin every talk I give with this phrase, breeding food for health. But what does that mean? Well, since the dawn of agriculture, humans have been breeding food crops for essentially one trait, yield. In our recent history, we begin to think about other traits, namely those concerned with nutrition. But before we can do that, we have to figure out what those traits are. At the Nebraska Food for Health Center, we seek to identify and characterize traits in plants that can improve human health, mediated by the human gut microbiome. Here's one way we do this. We take a staple crop, in this case maize, more specifically popcorn. We alter its genetics in such a way that changes the seed composition, in this case protein composition, that can in turn stimulate beneficial bacteria metabolite production in the gut microbiome that can ultimately improve human health. So, what is quality protein popcorn? Typically, 70% of protein in maize is comprised of zein storage proteins. These are large, relatively simple proteins that are deficient in the essential amino acids lysine and tryptophan. Quality protein popcorn is centered around a naturally occurring mutation, opaque-2, that shunts zein synthesis into other proteins, thereby completely altering the total protein composition and drastically increasing levels of tryptophan and lysine. Quality protein popcorn provides us with a unique opportunity to study the effects of radically differential proteomes within a single crop species on the human gut microbiome. And we do this by feeding a commercial variety of popcorn and the quality protein derivative to human gut microbiomes in vitro, allowing us to make direct comparisons between them in the microbiomes of 4 human subjects. Assessment by 16S sequencing and gas chromatography provide us with information on microbiome composition and metabolite production. The beta diversity analysis shows significant clustering of the microbiome samples treated with quality protein popcorn in blue and non-quality protein popcorn in red. We zoomed in on individual taxa driving this response and found something curious: an enrichment of several members of the Lactospiraceae family when treated with quality protein popcorn. Within this family, many species are thought to be probiotic because of their ability to produce butyrate, a pleiotropic, short-chain fatty acid associated with human health due to its role in appetite control and immune system regulation. We went on to test levels of butyrate in our fermentations and found significantly higher levels in the reactions treated with quality protein popcorn that correlated with genera within Lactospiraceae. Now, you may be scratching your head, much like we were. The production of butyrate by these organisms is almost always associated with fiber and carbohydrate fermentation, not protein. However, our inferred metagenomic enrichment identified pathways for the conversion of lysine to butyrate. Remember, only quality protein popcorn has lysine. And a further literature search identified another molecule that can be converted to butyrate by gut microbes. That is fructose lysine, which is a product of the Maillard reaction formed from lysine and glucose. And again, it's only present in popped quality protein popcorn, not the commercial varieties. To test our hypothesis, we fed these molecules to a microbiome in vitro and observed the same increase in butyrate we observed as a response to quality protein popcorn, indicating a probable mechanism of action. And not only did these treatments with lysine and fructose lysine stimulate butyrate production, we also observed different genera responding to different substrates. Coprococcus is an example of a genus enriched by quality protein popcorn, lysine, and fructose lysine, while Roseberia, enriched by quality protein popcorn, is apparently only able to utilize fructose lysine, indicating 2 avenues for microbial butyrate production from quality protein popcorn. This has been an example of a conventionally bred grain able to stimulate the microbiome in a positive way. We demonstrated this pipeline in popcorn, but it can be applied to any grain, to any plant, to begin improving the nutritional properties in respect to the human gut microbiome within staple crops that everyone has access to in a new generation of food for health.